Welcome to all, and thank you for attending this webinar. This will be about bringing innovation and performance through new delivery forms. We will introduce new products, StabiCaps and SiMoGel. My name is Pierre-Albert Thomas. I am the Global Technical Support Director at Rousselot. I will deliver an update on our solutions to overcome cross-linking in softgel with StabiCaps. I am here together with Dr. Jeff Delman, Business Development Manager at Rousselot. He will touch on the new concept of nutraceutical gummies with SiMoGel. Finally, Claude Capdepont, the head of our application laboratories, will answer the questions you might have. Very briefly about Rousselot Company. Rousselot is a brand of darling ingredients, and this is the global leader of collagen solutions, including gelatin. We supply solutions all over the world for health and nutrition, biomedical, food, pharmaceutical applications. In particular, we market a wide range of gelatins for pharmaceutical and nutraceuticals applications, and we hold a leading position in the segment of solid dosage forms. We estimate that more than 1 out of 4 gelatin capsules and soft gels in the world is made with Rousselot gelatin. This capsules market represents a consumption of 600 billion of capsules per year. In the recent years, we have seen the nutraceutical gummies segment emerge. Today, we will focus on the soft gels and the nutraceutical gummies applications. Soft gels are a highly popular solid dosage form, and in the world, 2,500 soft gels are taken every second. Those soft gels contain various types of fills that can be liquids, semi-solids, paste, even gels, and soft gels improve the bioavailability of the API. Gelatin soft gels provide the necessary protection for the API and beneficiate of very wide acceptance by the patients and the consumers. And this is because they are convenient to carry, easy to swallow, to identify as well with many possible colors. I don't want to spend too much time on the soft gel composition. Many of you are already familiar with the product. I will just say that soft gels are made of a shell, which contains gelatin and a plasticizer, and a fill. The fill will contain the dissolved or emulsified or even suspended API. In some very specific conditions, cross-linking may occur. This will result in a stability defect for the soft gels. This is a nightmare for new product development people. After having spent months or years formulating and testing, they realize during the stability tests that the capsules will not dissolve properly. Therefore, it's key to understand what triggers cross-linking and how we can predict the behavior of the formulation. A first mechanism corresponds to what I call the intrinsic cross-linking. Gelatin is a protein, and under stringent conditions of temperature and moisture, the carboxylic and amine group of the amino acids can react. This will trigger a cross-linking reaction that will make the soft gel become insoluble. Of course, the most severe cross-link reactions will occur in presence of chemical reagents. So they can be aldehydes, vitamins, minerals, polyphenols, specific APIs, also divalent cations, and so on. Most likely, those elements are part of the fill, but in some very few cases, I have seen elements from the shell react as well. In that case, they were dyes that were used in the shell formulation. So both the fields, which today become more and more complex, and the storage conditions will have an impact on the intensity of the cross-linking reaction. The choice of the right gelatin is therefore key. We know that some of the characteristics of the gelatin, such as its molecular weight distribution, might influence the cross-linking reaction. The pH as well might play a role. Predicting cross-linking and its intensity has always been one of the grails in the field of complex formulations with soft gels. We in Rousselot have worked on this topic, and we have been able to develop a rather quick predictive test. The principle of the test is to expose gelatin solution to a cross-linker and monitor the viscosity of the gelatin solution over time. When cross-linking occurs, the viscosity will increase exponentially. This is what you can see on the slide right now. And this test permits us to screen gelatins and select gelatins which are the least reactive to the cross-linkers. And about StabiCaps, this is a gelatin product that we developed. We performed many studies with our predictive test I just explained, but also in real conditions where we casted gelatin films and produced actual soft gels. So this StabiCaps range is based on lime-bound gelatins mainly, and includes StabiCaps and StabiCaps Plus. The difference is that StabiCaps Plus contains salts and has enhanced capabilities to overcome crosslinks. Both gelatins are different in viscosity and pH from the standard lime-bound gelatin, but they still procure the necessary machinability on the encapsulation devices. Now, back to our predictive test. You can see here that StabiCaps gelatins significantly delay the cross-linking reactions in the conditions of our test. And those conditions are very stringent. That's an exposure to a cross-linker, as I explained. And here in our studies, we have compared many different gelatins. Here on the slide, we have just an illustration with 5 different gelatins. But in all cases, StabiCaps and StabiCaps Plus outperforms all grades in the market. Ultimately, we want to confirm the performance of StabiCaps in real life. And we want to make sure that StabiCaps will improve the stability of the soft gel and the availability of the API. And to verify that, we have performed stability tests with various fields and various conditions over 6 months. What we did is actually mimic the soft gel shell by casting gelatin films. Then we applied on those films stringent storage conditions and also exposed them with specific fields that have been selected based on their cross-linking ability. And to determine which field we could use, we have cooperated with some of our customers. Here we have looked at the impact of what I called earlier the intrinsic crosslinks. And here we obtain a very clear result with StabiCaps having a superior performance versus standard gelatin. So here we have used stringent conditions of temperature and relative humidity to trigger those intrinsic crosslinks. In this next study, we have used a vitamin fill, which was identified as creating cross-linking issues. And we reached the same conclusion, that is to say that StabiCaps significantly improves the dissolution after 6 months. In this other study, we used a cranberry fill, and this fill as well was known to contain polyphenols that trigger cross-linking. And we were able to verify again the excellent performance of StabiCaps versus regular gelatin. By working on our pilot encapsulation machine, and also together with our customers, we have noticed some additional benefits of StabiCaps in terms of manufacturing. So these are not the primary benefits of StabiCaps, but it's always good to notice that we had a reduced maturation time and a faster drying of the soft gels. And we believe this is related to the specific molecular weight distribution of StabiCaps gelatins. About the impact of StabiCaps on formulation, there is a need to slightly adapt the soft gel formulation when working with StabiCaps. but these are minor adjustments. It basically means increasing slightly the gelatin content and reducing the water content in the recipe. And this is done to maintain the optimal gel mass, viscosity, and encapsulation. And this is what you can see on the curve on the right. By adjusting the gelatin content, we can mimic the regular viscosity curve of the traditional gel mass. Now, another important point is, of course, the regulation. And StabiCaps are just regular gelatins from a regulatory standpoint. They have the same level of compliance for food and pharma regulation than the other gelatins. In the case of StabiCaps Plus, which contains some salts, the regulation in the individual countries the product will be used has to be checked. This is also a patented technology. So earlier I have explained that StabiCaps range is based primarily on limebone gelatin, and as well we have developed a StabiCaps solution with similar performances with acid bone gelatin and hide gelatins. So this is the conclusion for the soft gels and StabiCaps part. Now I will leave Jeff explain you more about our solutions for nutraceutical gummies.
So thank you, Pierre-Albert, for that introduction, and good afternoon to all of you listening in. I'm Jeff Dahlman, Business Development Manager, Innovations at Rousselot. And before I start to talk about functional confectionery, I have 3 questions for you to answer. I'm going to go through them one by one, so I have time to choose. First question is, do you take one or more food supplements? Answer yes or no. Can be also more than one. Second question is, if you would have to take one every day, would you prefer a pill or a gummy? And third question, what do you think kids prefer, pills or gummies? So again, 2 options. Now, if you are a representative sample of the average population, about 75% of you will have answered yes on the first question. And that goes for almost all of the general population. There is little variation in terms of age, education, gender, income. On average, every 3 in 4 people take some kind of supplement. For the second question, it's a bit harder to find data, but I've only met few people who prefer tablets or capsules over gummies, and we'll find out later in the questions if you are the exception. And for the third question, I guess it goes without saying what kids prefer. So you may be wondering, gummies, is that still a big thing? Well, yes, gummies are still driving the market. Even in normal, non-functional sugar confectionery, 24% of the NPD is still in gummies, and 88% of that is in gelatin gummies alone. So roughly 1/5 of the total new product development in normal confectionery is in gelatin gummies. And that's knowing there are many other kinds of confectionery out there. There's popsicles, marshmallows, chewy candy, toffee. So it is very much a big thing in confectionery. But also, if you look at supplements or functional confectionery, we see the same thing. Gummies already are 10% of the supplement market. And even in the US, if you look at non-pill markets, it's 30% of the market. And unlike what you would expect, it's the majority for adults. Almost 2/3 is for adults. Now that brings me to my most important question of my talk. What is SiMoGel? Well, whenever customers or friends ask me, I tell them it's a solution for starch-free molding. And then they all go, without exception, so why is that important? And the answer to that question is basically my talk today. And I'll give you 2 reasons. But before we go into reasons, first, a bit of history. We go back to 1922 when a German confectioner invented a very clever way to make gummies. And to this day, they are still one of the world leaders in this sector. And the method he invented is still used widely by the entire industry, actually. And let's take a look at that method in detail. So you make gummies, you get your ingredients, which is basically sugar, gelatin, glucose syrup, water, flavor, color, and acid. You mix them, you cook them, and then you make them into a bear shape. Now, as you can obviously imagine, the key is make them into a bear shape. You have this hot liquid, about 80 degrees warm, that sticks to everything it touches. So how are you going to get this in its shape? So the key was and is to use starch molding. So let's take a look at this in detail. So like before, you get your ingredients, you make your slurry, and then you make a starch mold. This is basically a wooden or plastic tray filled with cornstarch. And in this starch, you create a bear-shaped cavity, and this cavity is then the mold where you deposit your slurry. Once the mold is filled, you take the trays offline and you put them into storage for 24 hours. During these 24 hours, the gelatin sets and the starch absorbs roughly 4 to 5% of the water still in the gummies. And after this storage, you separate the gummies from the starch, you clean the gummies, wax and package them, and the starch, well, the starch is physically cleaned. You remove any pieces of gummies remaining, you dry it, and you reuse it. So what is the problem with this production method? Well, if you're making normal gummies, there's nothing wrong with this. It's perfectly fine. But the story is more complex when you produce functional gummies. As an example, imagine you're making a batch of caffeine gummies first, and then you make a batch of vitamin gummies for kids. As a parent myself, I truly hope there are no caffeine traces in the kids' gummies, because if there are, that's going to be a sleepless night for me and my wife. But how do you make sure there is no cross-contamination? Because after all, the starch is dried and reused. So how can you make sure there are no traces left in that starch? And maybe even more important, in case of an audit, can you prove it? And how do you prove it? Because the mold has just been destroyed when you took out the gummies. So it's very difficult to establish absence of cross-contamination in this kind of process. And it's not just the mold. The starch is typically everywhere. If you've ever baked a cake or a pie, you know you always spill some of the flour on the kitchen counter. Even just closing the bag is messy. So imagine you have several metric tons of this starch going around the factory, around and around again. And as you can see in the pictures, there is a fine starch layer everywhere, on most of the equipment, on the trays, on the floor, next to the line. So, and again, I can't stress enough, this is perfectly fine if you're making normal food gummies, where there's no API or no functional ingredient. For these products, you just vacuum the dust regularly, and that's the end of the story. But in functional, or worse still, if you make truly pharmaceutical gummies, this is not a sight you want to see or show an auditor. So how does SiMoGel solve this? Well, we solve this by replacing the starch mold with a cleanable silicone or metal mold. Can even be a blister pack. So you make a similar slurry, deposit in the silicone molds, and then you let it gel for just 15 minutes. You then demold and you reuse the mold. Of course, when you switch products, you can easily send the silicone or metal mold to the washer. So that's one reason. SiMoGel greatly reduces the risk of cross-contamination in your production. And second, it's a question of scale. A starch molding line is called a starch mogul. So like a business mogul or a media mogul, it's used to signify it's big. And typically a line like this is easily 20 to 30 meters long. And a single line can produce anywhere from 1 to 5 metric tons of gummies per hour. And I haven't myself seen a small starch mogul yet. Now, this is great if you're producing a commodity product, if you're competing on price, then you want the biggest possible output at the lowest possible cost. But if you want to make premium functional products, maybe even personalized batches for your customers, do you really want to make 1 ton an hour? My guess is you prefer to make smaller batches, so you have more flexibility. And this is also what you see in the starchless lines. You can have a big line, like the one you see at the bottom here, but also smaller lines, like the one you see on top, doing 50 kilos or 200 kilos an hour. Now, I've explained you why you would use starchless molding for these products, but why would you need SiMoGel for that? And that is because time is of the essence. SiMoGel allows you to make the most of your starchless line by reducing the setting or the gelling time. If you look at a starch molding line, you can see at the left, you can see the trays here, and the trays are stored offline for 24 hours. So given that you have enough trays, the setting time doesn't impact the machine speed. But on a silicon line or metal molding line, the molds are fixed to the line. You see there's no stack of molds at either end. The molds just go round, which means that you have to empty the molds before you can refill them. That means the faster the gelatin sets or the gummy sets, the faster you can remove it from the mold and you can reuse it, so the higher your machine output is. With SiMoGel, we are able to bring the setting time down to as little as 15 minutes in silicon molds and even 6 minutes in metal molds. So that easily gives you a 4-fold increase in output. Now next we have a short video from some lines in operation. It's from FIE, where we did a live demonstration of this process. So here you see a small line in operation. It typically makes 50 kilos an hour, has about 8 pumps. And this is set up in trial for a bigger line. So this will typically make between half a ton to a ton per hour. In this case, they're using 3D molds, which is another advantage of this kind of technology. You can see the speed at which it's working. And then once they're dry, at the bottom, the molds open and the gummies get brushed out again. They fall into a cup below and they go for treatment at the next station. See strawberry-shaped gummies in this case. And this is just an example of the other more traditional molds, this time in metal. Now, in summary, what does Rousselot offer? We offer you the fastest setting solution on the market, but also our knowledge. We have 128 years of gelatin experience, and we provide full support from concept to finished product. We have an application lab where we have a lab scale depositor for testing concepts and improving recipes, but we can also do on-site production trials, and we have done in the past with existing lines, with new lines, so we know our solution works, and we can help you make yours work all the way from the concept to validation. And with that, it is now time to go to the Q&A. Thank you.
Good afternoon. I'm Claude Capdepont, the Application Lab Manager of Rousselot. So we receive questions, so I will start with the first question we received on SiMoGel. So about texture of SiMoGel, the texture of SiMoGel gummies are comparable to normal gelatin gummies. So we can observe in some conditions elastic texture, chewy texture, hard texture, but it can be comparable to what we have with gummies in general. So we can modify the texture with some solutions with other polysaccharides in the recipe. So that's what we can say about the texture of the gummies. We had some question also about the sugar-free SiMoGel. Yes, it's possible to produce SiMoGel sugar-free or sugar-reduced confectionery. We don't have specific challenges about this production because we can have exactly the same type of gummies at the end. The process is the same, using different types of polyol, different types of polydextrose, so it means fibers. soluble fibers, and we obtain the same type of gummies. So yes, it's possible to produce sugar-free and sugar-reduced confectionery. I have 2 or 3 questions about APIs in SiMoGel. Yes, we can use different types of APIs. Of course, we didn't test all APIs that we can find on the market, but we use a lot, and different vitamins, minerals, polyphenols can be used in SiMoGel. But of course, we have to test the other products that can be used also, and then it's something that we can develop further. I have a question about the raw material used for SiMoGel. So we have different types of raw materials. We have the bovine bone, we have the pig skin, we have the bovine hide raw materials. So everything is possible with in the range of SiMoGel products. I see now some question about StabiCaps. StabiCaps, the test protocol that we use, so it's an internal test that we develop. So we use a very strong cross-linker, which is aldehydes. So as Pierre Albert explained, it's a test that was developed in the application lab to be able to compare the different types of gelatin on the market, and we could rank it thanks to that protocol. This is a viscosity test, and we use as a cross-linker aldehydes, formaldehydes, and then we see clearly a strong difference between the different gelatin types. So it means that the different gelatin types react in different ways with the aldehydes. So then we could rank the product, and we could analyze our new development, StabiCaps, in that among these different types of products. We cannot say more about the test because it was developed internally for specific analysis in the lab. So if I look at the next questions, yes, we have about the protocol. So I explained the protocol to analyze the different, or to rank the property of the different gelatins versus StabiCaps. But we have also some tests, stability tests that we perform in the lab in different conditions. So that's what explained Pierre Albert. So we prepare films of gel mass and with the different types of gelatin, and we dip the films in different solutions of vitamins, minerals, polyphenols, and APIs that can contain very strong cross-linkers. And then we analyze the time after some weeks of stability. We analyze the dissolution of the product in the pharmacopoeia conditions. What is the advantage of StabiCaps over normal gelatin capsules? So Pierre Albert explained before that the design of the gelatin is a bit different because we have developed a product which is different in molecular weight. So then we have reduced the ability of a normal gelatin to cross-link. So then we have reduced the time necessary to cross-link in very strong conditions. So we never develop a modified gelatin. It's not the target. We don't have a anti-cross-link gelatin, but we have a reduced cross-link gelatin. So we have improved the capacity of gelatin to resist against cross-linkers. And then the cross-linker are different types of API containing sometimes aldehydes. So we have seen also that StabiCaps has an advantage compared to normal gelatin capsules, is that we can reduce also the drying steps because we increase the content of gelatin in the gel mass. That helps a lot to reduce the drying time. So we don't use a StabiCaps for any kind of filling, but we can use it in a case of very sensitive filling containing API that cross-link easily, or we can also improve the drying steps of the gel mass, normal gel mass. When you speak about the ages of StabiCaps after cooking or melting process, I suppose that you speak about the stability after the preparation of the gel mass. So normally, because you increase the concentration of gelatin, because the molecular weight of the gelatin is quite low compared to normal one, then you can use quite quickly the gel mass after the preparation of the gel mass. But we have tasted the product in different conditions, and we can also use it as a normal gel mass, and we can let it melt and let it stabilize for 4 or 5 days. That is possible too, because we can compare. But in that case, of course, we have increased the concentration of the gelatin in the gel mass. I've seen some question about hardness of soft caps. So we don't have any difference between gel mass, soft caps produced with StabiCaps and with normal gelatin. So the gelatin is a gelatin. StabiCaps is a gelatin. So even though the molecular weight is different, this is a gelatin with a normal property of the gelatin. So then the viscosity is the same, and then we can use it on the same condition than the normal gel mass. So then we don't have any difference in texture of the soft caps after production. So the temperature of gelling of SiMoGel, in fact, we can say that the SiMoGel sets very quickly. It starts to gel below, in fact, 30 degrees. In fact, is the reason why we can improve the setting. We have improved the setting properties, because very quickly after the production in silicone molds, we can remove the gummies from the silicone molds or from the metallic molds, and very quickly we will have the gelation of the gummies. So I see some question about vegan applications. We have to remind that gelatin, SiMoGel is a gelatin, and the gelatin is an animal product. So we cannot say that SiMoGel is a vegan product. So sorry for that, but vegan doesn't fit with gelatin. So for the I see about the formulation used for SiMoGel. So I see that to explain more about SiMoGel, SiMoGel is not only a gelatin, it's a concept. So for SiMoGel, we have developed a specific gelatin, but also a specific recipe. So we can we can give information about the formulation that we use, and then we can support you on the development of the SiMoGel, on the use of SiMoGel on your equipment. We can pack in blisters, of course, because of the fast setting property of SiMoGel. We can pack in blisters, in silicone molds, in metallic molds, where the fast setting is very important. And then we have, in that condition, a very fast property of the product in that conditions. read all the questions. What common nutraceutical products are marketed as gummies? So in fact, what we have tested in the lab are all the vitamins, the minerals, the polyphenols products. We can say that these are nutraceutical products because they are used in OTC. They are sold in OTC on the OTC market. Maybe Jeff, if you want to add more information about this market.
Well, thanks, Claude. I think if you look at common functional parts on the market, well, the bulk is probably still vitamins or multivitamins, but we're also seeing pre-workout gummies, post-workout gummies, even caffeine gummies are popping up. And if you look at the US, there's a lot of CBD and cannabis-based products used in gummies. So it's quite wide, but I would say for now the bulk is probably still just multivitamins. So back to you, Claude.
Okay, so I see a question about cross-linking with SiMoGel in gummies applications. In fact, SiMoGel is a gelatin. So we don't see any cross-linking in normal gelatin in gummies. So we don't see any cross-linking in SiMoGel applications. So we have answered almost all the questions about SiMoGel and StabiCaps. Then I think that now we can close the webinar sessions. I thank you all for all the interesting questions you asked, and thank you. Goodbye.